Good morning. In last practical, I have discussed to, I, to carry out isolation of pure culture by use of streak plate technique. Today we will see the next practical, that is to carry out the isolation of pure culture by the use of pour plate technique. So this is the aim of practical. Yes, what is the aim of practical? To carry out the isolation of culture by use of pure plate technique. We will see the requirement of this practical first. In that culture, the soil isolated culture E. coli and Staphylococcus aureus. In culture, the soil isolated culture, then E. coli, that is Escherichia coli, and Staphylococcus aureus. Then next chemical, that beef extract, yeast extract, then peptone, then sodium chloride, agar, and alcohol. Then next apparatus, conical flask, test tube, petri plates, micrometer loop, beaker, and test tube rack. And next equipments, the equipments required for this practical, that is autoclave, hot air oven, pH meter, balance, water bath, and thermometer. So this is all about the requirement. In culture, the soil isolated culture, then E. coli organism and Staphylococcus aureus. Then chemicals, peptone, beef extract, yeast extract, sodium chloride, agar, and alcohol. Then apparatus, conical flask, test tube, petri plates, micrometer loop, beaker, and test tube rack. Then equipments, autoclave, hot air oven, pH meter, balance, water bath, and thermometer. Then we will see the principle of this practical. In pour plate technique, the dilution of mixed culture is carried out directly in test tube of liquid agar medium. Yes, in this technique it means in pour plate technique, the dilution of mixed culture is carried out directly in the test tubes of liquid agar medium, and the medium is maintained in liquid state. The medium is maintained in liquid state by keeping media at high temperature to allow the proper distribution of the inoculum. To allow the proper distributions of the inoculum. The medium is then prepared into petri plates and allowed to solidify. Then incubate all plates under suitable condition. Then incubate all the plates under suitable conditions for growth of microorganism. Then next, a series of agar plate is showing the decreasing the number of colonies. The series of agar plate showing the decreasing the number of colonies resulting from the dilution technique. And if this technique is performed by serial dilution method, that is by using pipette, the counting of microbes are possible. Yes, in principle, if this technique is performed by the use of or by serial dilution method, that is by use of pipette, the counting of microbes are possible. So this is the principle of this practical. Next, advantages of this technique. First advantage of this pour plate technique, the microorganisms are uniformly spread in liquid agar. The microorganisms are uniformly spread in a liquid agar. Therefore, the microbes are also present below surface. The microorganisms are uniformly spread in liquid agar. Therefore, the microbes are also present below surface. Hence, the surface and subsurface colonies are developed. And it is very difficult to remove or transfer the subsurface colonies to the fresh media. Yes, disadvantages of this method technique. First, the microbes are uniformly spread in liquid agar. Therefore, the microbes are also present below surface. Hence, the surface and subsurface colonies are developed. It is very difficult to remove or transfer the surface colonies into the fresh media. Then second disadvantage, those microbes which are sensitive to higher temperature, which are sensitive to higher temperature, it cannot be isolated by pour plate technique because the temperature of agar should be always high. Agar should be always high to maintain at liquid consistency, and hence pour plate method is unsuitable for pour plate method is unsuitable for isolation of psychrophilic microorganism. Next we will see the procedure. First, prepare nutrient agar medium. And transfer 15 to 20 ml medium in each test tube. First, prepare nutrient agar medium and transfer 15 to 20 ml medium in each test tube. Then second, sterilize all test tubes in autoclave at 121 degrees Celsius at 15 IBS for 15 minutes. Then after sterilization, then cool all test tubes at 40 to 45 degrees Celsius temperature. Then after cooling, then simultaneously sterilized 3 Petri plates in a hot air oven and mark by using glass marker and mark as A, B and C. Then transfer one loopful of culture in a first test tube and gently mix the culture by rolling the tube between the palm. Then transfer one loopful of culture from first test tube A to second test tube, that is B, and mix the first inoculated tube. Then transfer one loopful of culture from second B to third test tube C and mix all stated in step 1. Then all inoculated liquid medium is poured into separate sterile Petri plate. All inoculated liquid medium is poured into separate sterile Petri plates. Then after the medium solidifies, place the Petri plates in a incubator at 30 to 35 degrees Celsius for 48 hours in an inverted position. So this is all about principle. Next, that is observations and result. The colonies appearing in Petri plate A are not distinct, small and overlap with one another. Then in Petri plate B, the colonies are large, discrete and generally do not mix with other colonies. And in Petri plate C, the colonies are larger and isolated colonies with distinct shape, color, texture, size and forms are observed. From Petri plate A to C, there is dilution of inoculum and only less number of microorganisms are allowed to grow in a Petri plate C. And colonies are developed on surface and the surface of the medium. So this is all about the observation and result. So what is observation result? The colonies are appeared in Petri plate A. These are not distinct, small and overlap with one another. Then in Petri plate B, the colonies are large, discrete and generally do not mix with other colonies. And in Petri plate C, the colonies are larger and isolated colonies with distinct shape, color, texture, size and form are observed. From plate A to C, from Petri plate A to Petri plate C, there is dilution of inoculum and only less number of microorganisms are allowed to grow in a Petri plate C. And the colonies are developed on the surface and subsurface for the medium. So this is all about this practical. We will see the questions based on this practical. First, name the any one collection Name the any one culture collection center, that is NC. Answer for this, that is NCBI, that is National Collection of Industrial Bacteria. I had already discussed this. Then next, what is pure culture? These are the culture which contain yes only one kind of these are the culture which contain only one type of microorganism, that is pure culture. Then what is mixed culture? These are the culture which contain more than one kind of microorganism, is called as mixed culture. Then next question, what is meant by isolation? It is the separation of microbes into single colony. Yes, what is isolation? It is the separation of microbes into single colony. Then how to preserve and how to maintain the pure culture? How to preserve and how to maintain the pure culture? By periodic transfer to fresh media, then overlaying the culture with mineral oil, or by the use of technique, that is lyophilization. Then next question, that is which type of solidifying agent is used? Which type of solidifying agent is used in streak plate technique for the preparation of medium? Agar is used as a solidifying agent in streak plate technique. Then next question, which technique is performed quantitatively to determine the number of bacteria of particular type? Then streak plate technique is used. Then next question, the psychrophilic microorganism cannot be isolated by which method? That is pour plate method. Then next question, reduced growth of obligate aerobes is a major issue in which technique of isolation? It is the major issue in pour plate technique. Answer for this, that is pour plate technique. Next question, which isolation technique is used to study the bacterial sensitivity to antibiotics? Spread plate technique is used. Answer for this, the spread plate technique is used to study the bacterial sensitivity to the antibiotic. So this is all about this practical, that is to carry out the isolation of culture by using pour plate technique. I will stop here. Next practical we will see in next practical.